exactly is structural biology, and what's involved in the day-to-day life of a structural biologist? Basically, structural biology is a subfield of biology that uses techniques like X-ray crystallography, cryo-EM, and NMR to get a look at proteins and other like macromolecules, so maybe things like DNA or RNA, maybe bound to proteins, maybe big complexes of them. Why? In order to try to figure out how what they look like connects to what they can do, in order to answer questions like, well, how do they do what they do? Can we get them to do it better? Can we design molecules that can interact with them? And so by solving the structures of these complexes and of these proteins and things, basically what we mean is we're figuring out the atomic positions of each of the atoms, or at least most of the atoms that make up those molecules, to try to like kind of get an X-ray of their insides, in order to figure out how they work. Just kind of like how you can see from a fork and a knife and a spoon how their form connects to their function, the same is true for biological molecules. And so by figuring out their form, we can better understand and figure out their function. But actually getting that structure is just a small part in the life of a structural biologist, at least typically. And a big part comes both before getting that structure and after that getting that structure. So there's a lot of kind of like optimization and things that go into trying to figure out the best way to purify that protein or that complex. Maybe you have to make some modifications to it before you can even get it to be in a form that's going to be amenable to these techniques. And then afterwards, there's a lot of follow-up to kind of characterize what you found and really verify it. If you were to place a hole in the base of like the bottom of a spoon, it wouldn't really make that much of an impact when you're eating your soup. But if you were to place a hole in the spoon part, now it would make a big impact. And so similarly, we can use the structures to kind of help guide the design of mutations and things to these proteins in order to figure out, like, is our hypothesis correct? Does that part really do what we do? Or does it just look like a spoon, but really it's not a spoon? Or it would be really bad for eating soup for some reason. Maybe there's like, I don't know. But anyway, basically there's a lot of biochemistry and like biophysical techniques. So things like we use to measure binding, as well as like measure activity and things like this. So there's a lot of like these activity things to verify what we found, as well as then there can be things like screening and various things to try to figure out molecules that interact, to try to really use the structure to help design things. So basically, there's a lot that goes into being a structural biologist, not just solving the structures, and that's just one part of it. So basically, you have to get the protein, get the complex, or whatever it is. You have to figure out a way to get a really pure version of it, typically, that is going to be stable enough for your cryo-EM, for your X-ray crystallography, for your NMR, whatever technique you're using to actually get a look. Then you have to optimize it. Not just does it have to be stable, but it has to actually be kind of cooperative when you're doing these experiments, and so you might need to do a bunch of troubleshooting for that. Then you have to figure out like what the structures look like. So after you collect all that data, you have to analyze all that data. And once you've analyzed all that data, well, now you have a bunch of theories, but now you have to go test those theories. And so then you go test those theories. Sometimes this is done in collaboration with other labs, especially if there's things that need to be done in cells and things like this. Often with structural biology, you're dealing typically almost exclusively with like in vitro work. So things like outside of the cells, taking a really reductionist approach, purifying all the components and mixing them or things like that in order to really get down at how they're working mechanistically. So we're kind of getting a look inside the gears of these proteins. And so then we want to make sure that when we're studying them, we can kind of break things down into their smallest parts. But then you lose a lot of the outside influence, which can influence things. And so then it's also important to study not just how these things work in isolation, but how they work in the bigger context. And so this is where like cell biology and things like this can come into play, often done through collaborations with other labs, but sometimes done by the structural biology lab as well. Often the structural biology lab will include not just people who are doing the actual structure solving, but also people who are doing those experiments, maybe even people who are doing the chemistry to design molecules that can interact with these. Maybe people doing cell biology, various things like this. And so it could be a really collaborative endeavor, solving the structures and then figuring out, well, how can we learn from those structures, and how can those structures inform what we're doing? How can what we're doing inform those structures? And yeah, so form and function. So get into some examples of how structural biology has been really, really useful. For example, helping design like insulin that works better for diabetics, helping design vaccines for the coronavirus, and various drugs and things like this. So structural biology has a lot of really important like translational aspects. So basically you learn something which makes it lets you do something that's going to benefit things directly, kind of have this direct applicational use, as well as more basic research trying to like figure out, well, how do molecules work, which can then later on lead to translational things. And it's also just really important to get a fundamental understanding of how things work. So structural biology can have both these basic and these translational applications, and we'll talk a little bit more about some examples of these, as well as kind of just what is involved with being a structural biologist, because it can seem like it's kind of obscure field, but it's really, really cool. So let's dive in.
So I'm going to talk most in terms of proteins, but structural biology can also be used to solve the structure of other sorts of molecules. And especially these days, with advances in things like cryo-EM, people are solving the structures of big complexes. So proteins bound to other proteins, bound to DNA, bound to RNA, all of these various things. But for simplicity's sake, I'm just going to be talking mainly in terms of proteins, and then just know that it can be used for other things as well as like complexes, multiple proteins and things like this. I'm just going to be talking typically in terms of single proteins because that's easier to discuss. And so basically, these proteins are going to have these unique structures because these proteins are going to be made up of unique combinations of amino acids, or these building blocks, these letters that are going to influence how these proteins fold up and therefore how they can function. And so you might have heard of like AlphaFold and things like this, basically these softwares that try to predict how a protein is going to fold based only on its sequence, based only on its which amino acids it has in what order. And so the software can do a lot, but it still has problems, and it doesn't have the context that goes into actually making these proteins in cells where they get help folding sometimes. So this makes them fold a little differently. They might get modifications inside of the cell. They might be bound to other things. They might be part of complexes, all of these various things where the structural biologists are able to figure things out, figure out the real structures, but the software can't do that, at least not yet. But AlphaFold and these other tools can serve as really important roles to try to kind of help get the design structures that could be actually then tested with these structural biology techniques, as well as help us build models for things. So these tools do come into play in structural biology as well as in the broader field. But the actual experimental determination of structures is still very, very crucial and very important. And so, yeah, so don't think that structural biology has been replaced by AlphaFold or things like this. And also remember that structural biology, well, actually getting that structure is just a small part. Then you have to figure out how does that structure actually relate to that function? Does that spoon really act like a spoon? Is it just an illusion? Can we design something that's going to interact with it better and things like this? But often what gets the most attention is these big flashy models. And so when you see one of these stick things, one of these ribbon things, what you're actually seeing is an atomic model. And what the scientists actually get is something messier. So whether it's cryo-EM or X-ray crystallography, what you're getting is some sort of map this meshy thing. So in crystallography, there's going to be an electron density map, and in cryo-EM, this is going to be like a Coulomb potential map. And I talk more about these in past posts I'll link to. But the basic thing you need to realize is that you're not directly getting these models. You're getting some sort of evidence of the location of these atoms, but then you have to actually place those atoms into the model. And the better your map is, the easier you can do this. And so this is where the idea of resolution comes in. Where basically the higher resolution, you're going to have like a tighter map. And higher resolution corresponds to lower numbers. More on this in another post, but it basically has to do with how close two things can be, where you can still tell they're separate things. So it's kind of like being able to measure something with in centimeter terms versus in meter terms. So smaller numbers are better, higher resolution, you get that crisper map you can build your model into. And so basically all I wanted to really show you here is that you're basically going to get some sort of more noisy data that then you have to build the atomic model into. And there's a lot of work that goes into this, a lot of computational work on the software side once you get this map, as well as a lot of work going into getting the protein or getting whatever it is into a form that can give you a good map. And so there's going to be a lot of work that goes into it. And so, for example, if you were to look in the supplementals for a cryo-EM paper, you can see that they're collecting like millions of particles, basically millions of little images, or lots of like thousands of these images, and then picking out like millions of these little particles within those images. So basically a lot of work. But it's really important. And then once you get those structures, you can do all sorts of really cool stuff to follow up with more functional experiments, trying to use a variety of mutations and things like this in order to figure out how what you saw really does correspond to how it functions. And so the techniques that we use to get how we see it, this is typically what gets all the attention with structural biology. Techniques like X-ray crystallography, where basically you shoot X-rays at a crystal. So basically you have all that protein kind of lined up in the same orientation. You shoot X-rays at it, you get this pretty pattern of spots, and then you work backwards from the spots, where we call this diffraction pattern, in order to get that map that then we can build that model into. With cryo-EM, you're basically taking a lot of little picture type things of this sample when it's embedded in this thin layer of like vitreous ice, so basically like glassy water. And then you collect a bunch of pictures, and then you try to figure out what was the molecules within them and try to like classify them, group them together, and average all those images. So I'm not going to go into the details here. I have the details in other posts. There's also things like NMR, which is good for little things that are really flexible. Things like hydrogen deuterium exchange mass spectrometry, or HDXMS, where basically you use a heavy version of water to kind of label more flexible parts of a protein. I actually use this in my PhD work. It was really cool. But that's another technique that you can use. But basically all these techniques, what they're doing is they're trying to give you information about the protein structure. So this information can be really useful. And so just a few examples, and then we'll get more into, well, how do you actually get to this step where you can make a structure, and then how can you build off of that structure? So for example, the coronavirus spike protein, this is the protein that you're probably all familiar with. It sticks out on the outside of the virus. It helps it bind to our cells through the ACE2 receptor. And this protein actually gets cut by protease, a protein cutter. And this is going to allow it to undergo this like dramatic shape shift, this conformational change that lets it merge its membrane, the viral membrane, with the cell membrane, and dump its contents into the cell so the virus can make more copies. Because it undergoes this dramatic shape shift, you kind of have like this pre-fusion conformation and the post-fusion conformation. And basically, if you want to be able to study the pre-fusion, it can be hard because it has the tendency to transform into this post-fusion state. And so scientists, well, when they were in the very early days, they were able to study this pre-fusion state from early on, because they knew from working on past viruses that they can make these mutations in a specific place in this protein to help stabilize it in the prefusion state. So basically, they can make these mutations that make it so that it's harder for it to undergo that conformational change. And when they made these mutations, and this is just happening, like they're just studying this protein in isolation. This isn't happening like in the virus or anything like this. They're just studying this protein in isolation, like putting the genetic instructions for making it into like insect cells and things like this in order to make lots and lots of the protein. We'll get much more into how we can make lots of protein. I mean, how that's a big part of structural biology, but they can make lots of this protein with a specific mutation that's going to stabilize it. And this allowed them very early on to get structures of the spike protein in that prefusion state. And this was really, really important because having those structures helped them understand how things like how this could bind to cells, how antibodies may or may not bind, things like this. And this stabilized conformation has actually been able to be used in vaccines like the Novavax vaccine, as well as other mRNA vaccines to basically stabilize it in the state that's going to be best for the immune system to recognize and make antibodies against this prefusion state, which is the one you want to prevent from binding to cells. And so they were able to do all of this because they had knowledge from past structures of what mutations you can make in what region in order to stabilize the protein in the prefusion state. And again, before you go into any sort of conspiracies, this is all happening in just like the isolated protein. And what you're doing is you're actually making it like less able to infect cells or anything. You're stabilizing it in the form that is going to be easiest for our immune system to kind of recognize and ward off. But this was all done with the help of structural biology. And yeah, so the McClellan lab was really at the forefront of helping in this fight against the coronavirus. Okay, what about other diseases? So we've been talking about the coronavirus, which is all this recent stuff, but actually the use of structural biology goes back to or is useful for like medicine, goes back to like the beginning of structural biology. And so in terms of X-ray crystallography, we really have to thank Dorothy Crowfoot Hodgkin, who was a real pioneer in this technique. And she saw the structure of many different things, but one of them was insulin. And this was like her crowning jewel. This is what she really, really wanted to do. And it wasn't the first structure she solved, but she finally did solve it. And insulin, basically, it's important to allow cells to let in glucose. And so with people with diabetes, they either don't make enough insulin in the case of type 1 diabetes, or their cells can't take in insulin, at least enough in the case of type 2 diabetes. And so basically they don't get enough of that sugar taken in. That sugar stays in their bloodstream and can cause problems. And so Dorothy Crowfoot Hodgkin was able to solve the structure of insulin, of that signaling molecule. And this was important for scientists when they then wanted to figure out, well, how can we make designer insulin that we can better use to treat diabetic patients? And so by figuring out how this insulin was formed, how it was stabilized, they could introduce destabilizing mutations in order to make faster acting insulin, as well as stabilizing mutations to make slower release insulin. And so they were able to do all this by knowing the structure of these compounds, or of this protein. Also, in addition to making changes to the protein, you can make changes to, or you can figure out how to get things to bind to that protein. So this is structure-aided drug design, where basically there are different methods, but if you have the structure, you can kind of design a drug, especially if you have an initial hit. You can design a drug that'll then better match the binding site in order to get it to bind more stably and things like this. So for example, going back to our coronavirus example, there's like this protein. Basically it's a protease, the main protease of this coronavirus. It makes this long polypeptide where basically it has a bunch of its proteins connected together, and then it has to cut them apart. And it does so using this MPRO. And so it's a target of some some drug design campaigns. And so there was this thing called like the COVID moonshot, where basically they started with a fragment screen. So you take a lot of little pieces and you solve the structure of like crystallography to solve the structure of these little pieces bound to the active site of this protease. And then they could like build off of those little parts that hit in order to make full molecules. So those were just a few examples of how structural biology can come into play. But in order to get all those structures, you have to do a lot of work typically. Okay, and so what's involved with this work? Well, often what you need is to do these techniques, you're going to need a lot of protein. And it's going to have to be really, really pure. I should mention that there are other techniques that's like cryo tomography and things like this, where you're actually looking at things inside of cells. You don't need to be like doing the purification and things like this. But for your classic structural biology, you're going to be needing a lot of these protein or this complex, and it's going to have to be really pure. For cryoEM, you can get away with a smaller amount of sample, but especially for X-ray crystallography, you're going to need a lot of sample, and it's going to have to be really pure. With cryoEM, you can kind of use the computer to kind of like weed out the junk. But with X-ray crystallography, it has to be really pure. And so sometimes it like purifies itself when it crystallizes, but it's not going to crystallize if it's not very pure typically. So in order for it to crystallize, you're going to have it really pure. And in order for it to crystallize, you're often going to have to kind of make modifications to the protein in order to get it to crystallize, because to crystallize, it has to kind of be more stiff and sturdy and less flexible. So you might have to cut off loopy bits and things like this. And this is one of the places where computational software can come in really helpful to try to identify which regions might be more loopy and things like this. What mutations can you make to make things more stable? But then you're going to have to get lots of the protein. And going back to how we can even make these mutations and how we can get the protein is this idea that there's this central dogma where you have the DNA gene for something gets transcribed, it makes an mRNA copy of it, and then that gets translated, it gets made into protein. It uses instructions to make the protein. And so we can make changes to that DNA sequence to make changes to the resulting protein. And we can stick that DNA sequence into cells such as bacterial cells or yeast cells, or even mammalian cells, and we can get those cells to make lots and lots of that protein for us. And then we often what we do is we like get them to make a version that has an affinity tag. So we basically stick a little bit of extra sequence on the end. It's going to put a little extra protein on the end. This is going to then allow us to purify it better. And so when we're purifying, we're typically using methods like protein chromatography, where we're taking advantage of things like that little tag. We can use beads or these little resin that are going to specifically bind to that tag. And we can keep our protein bound, wash off the other stuff, and then take our protein off or elute it. We can also take advantage of natural properties of the protein, like its charge and its size. And typically in structural biology, we're actually using multiple of these chromatography steps combined so that we can get things really, really pure. In other fields of biology, you might see just like a single step purification, but with structural biology, you're often doing a multi-step purification. And this purification is often going to take a lot of optimization. And so when you're a structural biologist, you're typically going to become an expert in purifying proteins, as well as expressing these proteins in different cell lines. So bacteria can be good for some things, and it's cheap and simple, but it doesn't like to fold all proteins. So you might have to branch out, try other types of cells, try other constructs, so basically other versions of the protein, make different mutations to it and things like this in order to try to get a protein, enough of the protein that you can then purify and study. And so all of this is going to start off typically, unless you're dealing with just the native protein, so that what the cells naturally make, if you're just taking it out of the cells directly, you don't have to do any sort of cloning. But typically you're going to be starting with some molecular cloning. And so when we talk about molecular cloning, this is basically just the process of getting that DNA gene, getting that information, well, typically it's like the cDNA, which is a DNA copy of the messenger RNA. I'm not going to get into that here, but basically you put the genetic instructions for making it. You have to get it into a form that's going to be able to get into the cells that you want to make the protein for you. And so you have to recombine your sequence with something like a plasmid, the circular piece of DNA that you can stick into the cells and use as like a vector or a vehicle for sticking it into the cells and getting the cells to make lots of it. And so there's a lot of molecular cloning that goes into play in the beginning of a structural biology project, typically, as well as throughout when you're making mutations to that sequence. In order to figure out what things are doing. And so cloning techniques include things like restriction cloning and SLIC, so sequence and ligation independent cloning, things like this. Often you're making lots and lots of constructs to try to see which works best. You're getting these cells to express this protein or this, and then you're purifying it. You're doing a lot of optimization, and then finally you can go about actually using those structural biology techniques to get a structure. And that structure, that's just going to be the first part. And so then you really want to follow up and see, okay, well, how does that structure really relate to its function and things like this? And how can we get more supporting evidence to the function of this protein or to support what we're seeing? So there are various other ways to study proteins. And so there are things that you can do to study the proteins inside of cells. With structural biology, the structural biology part, you're actually typically dealing with this recombinant expression. So we're making this protein in other cells. We can make this allows us to do things like make modification. We have the most control, but we lack the context. And so there's a bunch of experiments that you're going to want to do to try to show that, yes, this actually, what we're seeing really is physiologically relevant. It is biologically relevant. And so, like I said, this is often done in collaboration with other labs and that sort of thing. Not always, sometimes it's done by the lab itself. But there's a lot of work that goes into getting those structures. And so sometimes the expertise is different that's needed to deal with the structural biology work, as well as the functional characterization. But bottom line is there's going to be a lot of stuff that goes into getting that structure. And then there's going to be a lot of stuff that goes on afterwards in order to really kind of functionally test what you see. Yeah, so you're dealing typically with really reductionist approaches, trying to study things in their most purified manner in order to see how these molecules like work. And so you have the least like outside influence, which can be good or bad. So you're getting less distraction. You're not going to confuse like, oh, I think this protein is doing this, but really it's this other protein that was next door to it. That was doing what we saw. But you're not going to have the influence of that other protein, which might be modifying your protein or changing in some way how it acts. And also just because a protein can do something doesn't mean that it's normally what it does. So there's a lot of verification steps that need to go into play to really verify and validate what you find. And then once you find that, you often then go and make changes, and then you might actually want to get new structures. And so maybe you design a drug and you want to then find the structure of it, the protein now bound to that drug. Or maybe you want to make a mutation and then it stops the binding to the drug. And then you can show, like, using the structure, you can see why it no longer binds a drug. Or you can make some mutational change and then do some other follow-up, getting new structure. So it's a kind of circular process. Going back and forth between the structure and the function, and really trying to better exploit the relationship and understand the relationship. And so remember that there's going to be basically you have a lot of cloning, then you have the protein expression, optimization, purification, structure determination, and then functional characterization and going back and forth. In terms of the actual structure determination, this is often done with methods like X-ray crystallography and cryo-EM, NMR, HDXMS, which doesn't give you direct structure, but it can give you complementary information. And then there's a lot of computation that goes into actually figuring out the structure. There's a lot of it that can be done by computers, but then there's a lot of manual work that needs to be done as well, and making sure that all those amino acids are in the right conformation and things like this, the right shape, the right orientation in that model. So a lot of like work sitting at a computer and doing things, which is like my least favorite thing. But then you have that. Once you have that structure, then you can then do a bunch of experiments to further follow up. So we talk about like we talk about doing like biochemistry and biophysics and stuff. Basically, we're doing like actual experiments testing the function of these molecules, testing how they bind to other things, and how they act. As well as then you can do things like cell biology, maybe you do things like actually make the changes inside of cells and see if they have the same effect inside of cells, test various hypotheses and theories and things like this. So lots of really cool stuff that can come about because you get that structure. And so structural biology is this really, really cool key tool in the, or key like subfield or whatever of biology, but it can seem kind of archaic. So hopefully this post helped you better understand it, as well as some of the ways that it can come into use and what is involved if you actually want to be a structural biologist. Okay, so hope that helped.